Welcome to a journey behind the Iron Curtain, where a remarkable Cold War-era medical program has been quietly saving lives for nearly a century. While Western medicine embraced antibiotics, Soviet scientists perfected a fundamentally different approach to fighting bacterial infections. Today, we'll explore the fascinating world of bacteriophage therapy, a treatment using viruses that target bacteria with sniper-like precision, and how this forgotten Soviet science might be our best hope against the growing antibiotic resistance crisis. Deep in the laboratories of Tbilisi, Georgia, scientists at the Eliava Institute have been developing and perfecting bacteriophage therapy since the 1920s. Bacteriophages, or phages for short, are viruses that exclusively infect and kill bacteria. Unlike broad-spectrum antibiotics that indiscriminately attack all bacteria in your body, both harmful and beneficial, phages are exquisitely specific. They target only particular bacterial species or even specific strains, leaving your beneficial microbiota completely untouched. This precision represents a fundamentally different paradigm of antimicrobial treatment that works with rather than against the complexity of the human microbiome. As Dr. Mzia Kutatladze, director of the Eliava Institute explains, when penicillin emerged in the 1940s, Western medicine essentially abandoned phage research. But behind the Iron Curtain, necessity drove innovation. Without reliable access to antibiotics, Soviet scientists continued developing phage therapies for 7 decades. This divergence in scientific trajectories represents one of the Cold War's most consequential yet underappreciated effects on medical development. The isolation between East and West was nearly complete during the Cold War. While Soviet scientists published extensively in their journals, language barriers and limited scientific exchange meant Western researchers remained largely unaware of the sophisticated phage therapy programs evolving in Eastern Europe. This parallel development created 2 entirely different approaches to infectious disease, one reliant on chemical antibiotics manufactured by pharmaceutical companies, the other on biological viruses that had co-evolved with bacteria for billions of years. The Eliava Institute houses what might be the world's most valuable viral collection, over 800 characterized phage strains effective against dozens of bacterial pathogens. During the Soviet era, the institute produced tons of phage preparations annually, treating millions of patients for conditions ranging from dysentery to surgical infections. This industrial-scale application of phage therapy has no parallel in Western medicine, where phages remained primarily research curiosities rather than mainstream treatments. What makes the Eliava approach particularly fascinating is its adaptive nature. The institute regularly updates its phage cocktails by isolating new phages from sewage samples, ensuring effectiveness against evolving bacterial threats. This dynamic, responsive approach starkly contrasts with the static nature of conventional antibiotic development. The methodology developed at Eliava represents a fundamentally different relationship with microbial ecology. Rather than developing new synthetic compounds in laboratories, phage researchers work with natural selection, harnessing evolutionary processes to counter bacterial adaptation. When bacterial resistance emerges, researchers collect environmental samples from locations where resistant bacteria are likely present, sewage systems, hospital effluent, or polluted rivers. Within these environments, phages have naturally evolved to counter the resistant bacteria through the ongoing evolutionary arms race that has shaped microbial interactions for eons. This ecological approach extends to treatment protocols as well. The Eliava Institute pioneered phage cocktails, combinations of multiple phage strains targeting different receptors on bacterial surfaces, dramatically reducing the likelihood of resistance development. Some of their preparations contain dozens of phages, creating redundant killing mechanisms that bacteria find nearly impossible to evade simultaneously. As antibiotic resistance becomes a global crisis, Western researchers are now turning to this long-ignored Soviet research. A 2020 study in Nature Microbiology by Schooley and colleagues documented the successful use of personalized phage therapy to treat a multi-drug-resistant Acinetobacter baumannii infection in a critically ill patient, a breakthrough that would have been routine in Tbilisi decades ago. The renaissance is accelerating. In 2019, the University of California, San Diego established the Center for Innovative Phage Applications and Therapeutics, IPATH, North America's first dedicated phage therapy center.
Meanwhile, Belgian researchers at the Queen Astrid Military Hospital have treated over 400 patients with custom phage preparations through a compassionate use program.
This revival is particularly notable because it represents a scientific homecoming of sorts. Félix Darel, who co-discovered bacteriophages at the Pasteur Institute in 1917, initially attempted to develop phage therapies in the West. He collaborated with Georgian scientist Georgi Eliava to establish the Tbilisi Institute in 1923. While Darel's work was largely abandoned in the West after antibiotics emerged, his scientific descendants in Georgia maintained and expanded his vision for nearly a century. The modern reappraisal of phage therapy coincides with our evolving understanding of the human microbiome. As researchers recognize the collateral damage that broad-spectrum antibiotics inflict on beneficial bacteria, the precision of phages becomes increasingly attractive. Studies at Stanford University have demonstrated that antibiotic treatments can disrupt gut microbiota for up to a year after treatment, potentially contributing to conditions ranging from inflammatory bowel disease to metabolic disorders, side effects largely absent in phage therapy.
The implications extend far beyond treating infections.
Recent research from the Weizmann Institute in Israel revealed that phages interact with mammalian immune systems in previously unimagined ways, potentially modulating inflammatory responses in conditions like inflammatory bowel disease. Even more surprisingly, microbiologists at the University of Leicester discovered in 2021 that certain phages can disrupt bacterial biofilms, complex bacterial communities protected by extracellular matrices, making them virtually impenetrable to antibiotics. This finding has profound implications for treating chronic infections associated with medical implants and cystic fibrosis. The phage-human relationship appears increasingly complex and important. A 2022 metagenomic analysis by San Diego State University researchers identified over 10,000 previously unknown phage types residing in the human gut, suggesting that phages may be integral, not incidental, to human health. Some of these phages appear to preferentially target pathogenic bacteria while leaving beneficial species untouched, suggesting a form of evolved symbiosis between phages and humans.
This symbiotic relationship might even extend to neurological function.
Preliminary research from the University of Leuven in Belgium has identified correlations between gut phage populations and neurotransmitter production, opening speculative but intriguing possibilities that phages might influence the gut-brain axis. If confirmed, this would suggest that the virome, the collection of viruses in and on our bodies, may be as important to human health as the better-studied bacterial microbiome.
What makes phage therapy particularly remarkable is its biophysical elegance.
Using cryo-electron microscopy, researchers at the University of Jyväskylä in Finland recently visualized how phages dock with lock and key precision with bacterial receptors. This specificity explains why phages rarely disrupt beneficial microbiota, a significant advantage over broad-spectrum antibiotics. As Dr. Elizabeth Cutter, a pioneering phage researcher at Evergreen State College notes, phages are essentially self-replicating, self-limiting antimicrobials. They multiply only where their bacterial targets are present, then disappear when the infection is cleared. The evolutionary sophistication of phages extends to their infection mechanisms. Many phages have evolved enzyme systems that can degrade bacterial biofilms, allowing them to access bacteria hiding within these protective structures. This capability, absent in conventional antibiotics, makes phages uniquely effective against chronic infections that establish biofilm fortresses in human tissues.
The pharmaceutical implications are profound.
While antibiotics typically require multiple doses to maintain adequate concentrations in the body, phages amplify at the infection site, increasing as they kill bacteria until the infection is cleared. This auto-dosing effect means that theoretically, a single effective dose might be sufficient for treatment, a pharmacokinetic profile unlike any conventional antimicrobial.
Despite compelling evidence for their effectiveness, regulatory frameworks in Western countries remain poorly adapted to phage therapeutics.
Unlike conventional drugs with fixed chemical compositions, adequate phage preparations are dynamic, often containing multiple phages that may be updated regularly to combat resistance. This regulatory mismatch reveals how deeply cultural assumptions influence scientific progress. The Soviet approach, pragmatic, adaptive, and focused on outcomes rather than standardization, contrasts sharply with Western pharmaceutical paradigms, which prioritize consistency and intellectual property protection.
The intellectual property challenges are particularly significant.
Natural phages cannot be patented, and the continuous updating of phage cocktails creates a moving target incompatible with traditional drug approval processes. This economic reality has deterred significant pharmaceutical investment, despite the enormous potential market for alternatives to failing antibiotics. The story of phage therapy illustrates how geopolitical divisions can fragment scientific knowledge with real consequences for human health. For decades, the Iron Curtain prevented the cross-pollination of medical approaches, with Western medicine largely unaware of the sophisticated phage programs developing in Georgia and Russia. As antibiotic resistance threatens to return us to a pre-antibiotic era, this Cold War relic may become central to 21st century medicine. The convergence of Georgian phage expertise with Western biotechnology and regulatory frameworks could finally realize the potential of a therapy that has waited nearly a century for its global recognition. The path forward likely involves hybrid approaches that combine the best of both scientific traditions. Researchers at ETH Zurich are now using CRISPR technology to engineer phages with expanded host ranges and enhanced killing efficiency, while maintaining their inherent safety and specificity. Meanwhile, the European Medicines Agency has begun developing new regulatory frameworks designed explicitly for phage therapeutics, acknowledging their fundamental differences from conventional pharmaceuticals. As Dr. Kutatlads reflects, sometimes the most promising solutions aren't found in the newest technologies, but in approaches that were simply developed in places we weren't looking. In the case of phage therapy, looking beyond the historical divisions of the Cold War may provide one of our most potent weapons against the growing antibiotic resistance crisis. By combining the century of experience from Georgian phage researchers with cutting-edge Western biotechnology, we may find ourselves with a powerful new-old approach to fighting bacterial infections in an era where antibiotics are increasingly failing. The story of bacteriophage therapy represents one of medicine's most remarkable ironies, a potentially life-saving treatment developed extensively in the Soviet Union while remaining virtually unknown in the West for decades. As we face a growing antibiotic resistance crisis, this Cold War scientific divide becomes more than historical curiosity. It represents a critical missed opportunity that we're only now beginning to rectify. The Eliava Institute's century of experience with phages offers a fundamentally different approach to bacterial infections, one that works with natural ecology rather than against it, that targets pathogens with precision rather than carpet bombing our microbiome, and that adapts to bacterial evolution rather than falling behind in the resistance arms race. As Western and Eastern scientific traditions finally converge around this approach, we may find that the solution to one of modern medicine's greatest threats has been hiding in plain sight all along, behind the geopolitical divisions that once separated our scientific communities.
In bacteriophages, we see not just a medical technology, but a profound lesson about how cultural and political factors shape scientific progress, and how sometimes, the most promising future comes from rediscovering a forgotten past. Thanks for watching.
Please like, subscribe, and hit the bell icon for updates.
Your feedback is appreciated. We hope to see you again.